Hello everyone, I'm Alzraa Mohamed from Zagazig University, Egypt. I'm a PhD student in microbiology. Also, I'm a content creator on YouTube. I'm happy to participate for the second time in this amazing conference. Actually, I'm interested in antimicrobials. The last year I participated in this conference with a presentation, a live presentation titled Nanotail and Antibiotic Resistance, Characterization of our Biocene Activity Against Gram-Positive Pathogens. Today, I'm sharing with you a new research related to antimicrobials about the combination of meropenem and zinc oxide nanoparticles, the efficient antibiofilm and therapeutic activity against bacterial keratitis. The published article titled Combination of Meropenem and Zinc Oxide Nanoparticles, the Antimicrobial Synergism, Exaggerated Antibiofilm Activity, and Efficient Therapeutic Strategy Against Bacterial Keratitis. The article was published in October 2022 in the Journal of Antibiotics in MDBI. I'd like to thank my colleagues and co-authors. Also, we would like to thank the Deanship of Scientific Research at the King Abdulaziz University in Jeddah, Saudi Arabia for supporting this work under grant number RG 1416642. Before talking about the new findings and the results of our research, I'd like to share with you the motivation and the background behind this research. What we see on the left hand is a summary of the World Health Organization's global report in 2014 about the antimicrobial resistance around the world. It's expected that the antimicrobial resistance will kill about 10 million people annually by 2050. However, on the right hand, we see a summary of a study published in February 2022 about the bacterial AMR in 2019. As we see in this summary, the bacterial antimicrobial resistance caused from 3 to 6 million deaths associated with antimicrobial resistance. However, the attributable number of deaths due to bacterial AMR was exceeding more than 1 million only in 2019. According to this study, there was an indicated 6 leading pathogens for deaths associated with resistance. Those 6 leading pathogens caused about 1 million of deaths attributable to the antimicrobial resistance and more than 3 millions of deaths associated with antimicrobial resistance. Among those 6 leading pathogens, P. aeruginosa was one of the antimicrobial resistance infections that caused deaths. P. aeruginosa able to produce extracellular polymeric matrix, which is called biofilm. The biofilm protects the bacteria from antibiotics. This makes it difficult to treat such biofilm infections. The biofilm enables the bacterium to develop chronic infections. As we see in this diagram, we see how antibiotic find it hard to penetrate the biofilm matrix to reach the bacterial cells. As we see in this slide, we see how the biofilm infections of P. aeruginosa are variable and severe. P. aeruginosa biofilm infections can affect the lungs in the cystic fibrosis patients, can affect wounds, can affect eyes in causing keratitis. Also, P. aeruginosa can form biofilms in the medical devices like catheters and other devices. So such antimicrobial resistance health threat moves us as researchers and everyone related to the healthcare systems to find solutions for such problem. Solutions for such health threat include two main approaches. The first is developing new antimicrobials and surveillance on the use of antimicrobials. Regarding surveillance or monitoring the antimicrobial use, the World Health Organization sets different approaches and strategies to follow up the consumption of antibiotics in healthcare units and to follow up the development of antibiotic resistance in all countries around the world. Developing new antimicrobials could take place in two ways. To find new antimicrobials like natural products from plants or from microorganisms or synthetic compounds, or the other route for finding or developing new antimicrobials is to modify and improve the inhibitory action of the currently available antibiotics using modifications of the structure or combinations. One of the most common and efficient combinations is nanoparticles to be combined with antibiotics, and it's known for its antibacterial activity, especially the metal oxide nanoparticles. Since we focused in this research on the biofilms of Pseudomonas aeruginosa, we aimed at using the zinc oxide nanoparticles to inhibit the biofilms of P. aeruginosa. Here we see how the nanoparticles, its small size enables it to penetrate the biofilm matrix and migrate through it to reach the bacterial cells within the biofilm to kill it. So it's a good strategy to use nanoparticles in combination or alone for treating or eliminating the biofilms. Here we move to the results of our research. In this work, we made this study on 24 P. aeruginosa strains. When we tested the antibiotic susceptibility of these bacterial strains, we found that almost all the strains was resistant to most of the used antibiotics. However, the Meropenem and Imipenem was among the highest resistance. We found all strains were resistant to Meropenem, and more than 91% of the strains were resistant to Imipenem. As you may know, Imipenem and Meropenem are the most used carbapenem pseudomonal antibiotics, and it's commonly used in the medical field. So when we found such resistance percent, it's important to work in these results. Therefore, we continued our studies on the Meropenem to find whether the combination of zinc oxide nanoparticles with Meropenem able to reduce this resistance rate, or how we could improve its efficiency. Before combining the zinc oxide nanoparticles with Meropenem, we tested the antibiofilm activity of zinc oxide nanoparticles against the biofilms formed by the 24 Pseudomonas aeruginosa isolates. As we see here in this figure, the green columns showing the untreated biofilm and the white columns showing the zinc oxide nanoparticles treated biofilms. As we see, the biomass of the biofilms was decreased about 70 to 85%, meaning that the zinc oxide nanoparticles has a powerful antibiofilm activity. Therefore, we continue the further studies to determine the minimum inhibitory concentration of zinc oxide nanoparticles alone and in combination with Meropenem. Here we see zinc oxide nanoparticles, the MIC, decreased from 64 micrograms to only 8 micrograms, and the Meropenem decreased from 512 micrograms to 4 micrograms. This is a great decrease in the concentration when we used a combination of both. When we calculated the fractional inhibitory concentration of each one alone and in combination, we found that the fractional inhibitory concentration index was less than 0.5, meaning that the addition of zinc oxide nanoparticle enhances the antimicrobial activity and works synergistically with the activity of Meropenem. Afterward, we examined the activity of zinc oxide nanoparticles against the P. aeruginosa biofilms microscopically using the scanning electron microscopy and macroscopically using the Congo-red agar assay. On the left, we see two plates, the dark plate showing how the P. aeruginosa was able to change the color of the medium from red to black, indicating formation of biofilm. But when we treated the P. aeruginosa with zinc oxide nanoparticles before inoculation on the Congo-red agar plate, it wasn't able to change the color of the medium, so the plate stays red. On the right, we see the scanning electron microscopic examination of the biofilms of P. aeruginosa. In the two upper plates, we see a high density of cells. In the middle plate, we see the zinc oxide nanoparticles treated biofilm. The cell density decreased somewhat. However, when we use the combination in the lower plates, we see significantly the number of cells has decreased, which meaning the powerful anti-biofilm activity of this combination, zinc oxide nanoparticles and meropenem. As we know, the biofilm formation by Pseudomonas aeruginosa is related to the expression of biofilm genes. These genes are the LAS-R, Fly-C, and BACIL-A. In our study, we tested the effect of zinc oxide nanoparticles on the relative gene expression of the biofilm genes, and we represented the data of each gene relative to the 16S rRNA. It's obvious that the expression of the tested genes decreased significantly in the treated samples when compared to the untreated samples. Also, about 2 to 4-fold reduction in the gene expression was observed after the treatment with zinc oxide nanoparticles, meaning that the zinc oxide nanoparticles work also on reducing the activity or the expression of the biofilm genes. We supported our work with an in vivo study of the activity of the zinc oxide nanoparticles alone and in combination with meropenem against the bacterial keratitis using a rat model. As we see in this picture, the black areas indicating or referring to the infection intensity, how the infection intensity changed and how it has decreased upon the treatment with zinc oxide nanoparticles alone and meropenem alone and the combination. According to the finding of this study, the P. aeruginosa recorded elevated antibiotic resistance against the tested antibiotics. Also, the antibiotic resistance increases the severity of the P. aeruginosa biofilm infections, which motivates us as researchers to find solutions or antimicrobial agents. In this study, we used zinc oxide nanoparticles with size less than 50 nanometers, and the small size of zinc oxide nanoparticles increases their ability to penetrate the biofilm matrix, leading to more enhanced antibiofilm activity, and this explains the complete removal of biofilm in some fields during the scanning electron microscopic examination. We found that the mixture of meropenem and zinc oxide nanoparticles downregulates the biofilm gene expression, which provides a reasonable explanation for the reduced antibiofilm density. The synergistic antimicrobial and antibiofilm effect of meropenem and zinc oxide nanoparticles combination successfully treated the cornea rat model from P. aeruginosa induced keratitis, which indicates the clinical significance of this combination, especially against bacterial keratitis. From this study, we have 3 recommendations. The first one, it's important to develop and find new antimicrobial agents to stop the growing antimicrobial resistance health threat. The metal oxide nanoparticles are successful candidates for improving the inhibitory action of the antibiotics. Therefore, we recommend researchers to study other metal nanoparticles for their potential synergism with other antibiotics for treating different infections. I hope you enjoyed this presentation and found it useful. I'll be happy to answer comments, any question in the comments below this video, and thank you for your